I'd say, what would you say are the most prevalent concerns, right? That sort of pervade your delivery vehicle, right? What are some of the perhaps challenges you face, right? Or you think that the industry perceptions that you think the industry has about this delivery mechanism that makes it a little more difficult for them to invest in, and what do we need to do as an industry to really kind of set the story straight, right, and get folks equally excited and pushing for alternative delivery vehicles such as the ones you're working on today. Um, let's start with Gloria.
Okay, sure. So the I mean, one of the things that we have um, looked at that is difficult is GMP manufacture in a very efficient and reproducible way, right? So when we're
Okay.
working with cells that produce from donors, they produce different amounts of things. And the flip side of that is that the regulatory agencies are quite familiar with this. These cell type therapies have been around for 30 years, as opposed to
Hmm.
these newer modalities, which are more get a little bit more scrutiny in terms of safety and long-term follow-up and what you need to show to be able to get into the clinic. So I
Hmm.
think that's one of the things that we really need to get better at is scaling up, into bioreactors and ways that we can make these at
Hmm.
a very large scale in order to be able to use them systemically. So most of what we do at this point is local delivery. So we're not working a
Mm.
lot
Yep.
systemically, but we have, interesting graft versus host disease results in mice, and how do we translate that into humans with a much larger scale manufacturing? So
Hmm.
that's one. The second is the perception that, and it's out there. And I used to work in wheat proteins, and everybody thinks wheat proteins, cause cancer, and others think they fix cancer. And it's a little bit of the same sort of legend with EVs and exosomes, is that they've gotten this idea that they can perpetuate cancer, and
Mm.
that I think that came from some animal models where people were using exosomes or extracellular vesicles from cancer animals and putting them in other animals. And yes, they could transmit the cancer, but we don't use donors that have cancer. So
Yeah. We're talking healthy
it's
donors here.
really a
Yeah.
non, we've seen absolutely no evidence that there's any potentiation of any kind of cancer in any of our animal models. And we actually, in the EV world, we have a lot of patients that develop squamous cell carcinoma regularly, because we have adults and they get them every year. They'll get a squamous cell carcinoma, and it doesn't hurt them at all. I mean, it has
Mm.
absolutely no effect on the course of their disease. So that's just another thing that we just need to address the
Hmm.
perception.
Yeah, absolutely. So it's thinking through. I really liked. Obviously, the one is industrializing, right? The manufacturing paradigm, being able to have more potential partners, right, to help develop this. Again, thinking on the CDMO landscape, right? Thinking through
Mhm.
getting a little bit more industrialized, as well as perhaps even more publications on the clinical side, right? More clinical data, I think, helps as well to dispel erroneous ideas, right?
Right.
Yeah. Melissa, how about you? What are some of the biggest, I'd say,
Yeah,
hurdles in perspective?
I would echo what Gloria said about the manufacturing. I think
Mhm.
that anytime you talk about a platform technology that utilizes cell-based manufacturing, there's just a groan that goes through the room. Right? You're like, oh, okay, it's not synthetic.
Yeah. Especially, I think,
And
in the RNA space too, right? Where we're so used to just everything's synthetic, right? So
yes,
it's like,
we're
ah.
why the LNPs are there, right?
Yeah.
And I do think, right, there are unique challenges to cell-based manufacturing, but again, they are solvable. And
Mhm.
I think a big piece of the challenge that comes down to these technologies is the perception of the COGS and the perception of
Mm.
cost,
Okay.
ultimately, of what your drug is going to cost and what you're going to be able to price.
Mhm.
Um, and I think a lot of the assumptions are based off of, you see price tags for some of these therapies that
Yeah.
utilize like lentiviral vectors as an example.
Mhm.
And I think a lot of the presumption is that
Mhm.
it's fully driven by COGS, and that's just not how we price drugs in the United States. So I think that there is probably a overestimate of what the costs actually are going into the production of these types of technologies that I think is a huge challenge that we have to continually overcome.
Perfect. Yeah, I think that's across the advanced therapy space, even in the LNP world, I think too, that continues to be a concern, especially for redosing. So, all right. So we've got cost of goods. We've got challenges here with the sort of industrialization of certain manufacturing paradigms. Sean, what are some of the biggest, perhaps misperceptions or challenges you're facing that are the most challenging for you to overcome, and what does the industry have to do? Right.
Yeah, I mean, I think from the regulatory stance, I mean, it's, I think from a non-viral is that a lot of people look at, clinical precedent, right? And in terms of LNPs have had a lot of clinical precedent, which has made regulatory barriers a little bit lower. But that said, how we've been looking on the polymer side is that a lot of the class of polymers that we use and explore are widely well characterized and actually have seen some in different applications within the clinical applications. So that's been allowing us to kind of let's say get ahead of the curve on the clinical side. But I think primarily is as I think everyone has touched upon is that there's definitely regulatory answers that you have, or regulatory questions that you have to answer technically, right? As to get people more and more comfortable, especially with new platforms, right? And so that just requires building more data packages, more GLP tox studies, making sure that you have translations from mouse to primate to get people kind of comfortable around a new platform.
Well, we do have a question here too, as well as what does the regulatory path look like for novel polymers? So do we have a regulatory precedent that we can kind of follow, or are we paving that path today?
Yeah, it's a great question. I mean, I think it's, how we've been looking at it is we're kind of, off the coattails of the LNP space, because there's not chemically they're distinct, but there's a lot of overlap between the two platforms. And so that's how we've been approaching it. The interesting thing about polymers is that they're effectively just they're one chemical entity, right? And so where you have lipids typically are four distinct, just operating at different ratios, right? And so you can make the argument that polymers are more chemically defined, right, in that respect. So it could be easier for regulatory, or people to get their arms around that. But that's how we've been going about it. We've been looking to the LNP space. Also, is that once we get in the clinic with a polymer that's approved, then you get a platform designation to then actually broaden that across indications. So.
Okay, perfect. And then Jim, tell us what are some of the biggest challenges or pushback that you might face in your world, and where does the industry need to kind of combat that, or how does the industry need to combat it?
Yeah, absolutely. I think the biggest challenge or misconception that the industry has in terms of cell-specific delivery is believing that the GalNAc and its receptor pair ASGPR are the only system that can deliver that way. That system has been extraordinarily successful.
Yeah.
7 FDA approvals based on that system, and now 100 plus therapeutics in development. And I hear all the time, oh, that must be the only system that does that. ASGPR is the only highly expressed receptor that internalizes. And if you know anything about receptor biology, all receptors internalize. And it's just a question. There are over 3,000 receptors in the human body. It's a question of saying, okay, how do we sort through this really broad target landscape to find the best receptor target for a given indication for a given cell type? And in our work, we've shown for over a dozen receptors that we can deliver cells specifically outside of the liver. We think there's a huge opportunity beyond the dozen that we're working on, potentially hundreds of receptors. And so, we really, from our perspective, we want more of the field thinking about how to leverage receptor thinking in the delivery platforms.
So what would that look like for the industry, right? Like what kind of investments are necessary to get this kind of information? Like what do folks have to do? Obviously, besides working with you, right? Besides working with Gnosis Bio, but what are some of the, biggest investments or types of knowledge we need to be digging into more? And who's responsible for that, right? Who needs to do that?
Absolutely. Well, we have, if you look to the small molecule or monoclonal antibody space, we have these incredible target discovery and validation platforms for finding the absolute best target for any indication that's druggable. But I would say the RNA field has not really developed that capability set for around delivery. Delivery is really came from chemistry and formulation. It did not traditionally come from biology. And so we think more biologists need to be involved in thinking about delivery and really build the target discovery and validation platforms. And it's different assays, it's different data sets, it's different work that needs to be done in labs, but there's a clear roadmap for doing it.